Good morning, how are you? Today, inshallah, we will start with lesson 2 in chapter 10, which is the process of cell division. You have to know what we mean by cell division, what's the ratio of surface area to the volume mean, what will happen to the surface area to the volume ratio once the cell grow, okay? For today, inshallah, we will start with regulating the cell cycle and how the cell cycle will be regulated in both prokaryotic and eukaryotic cells. Okay, let's classify first the cells. In the previous chapters, we classified the cells into two types according to the presence of the nucleus into prokaryotic and eukaryotics. Once we say prokaryotics, it means there is no true nucleus. And in eukaryotic, there is a true nucleus within nuclear membrane. Here in the prokaryotic cells, there is no nucleus. It means the DNA will be in the cytoplasm. While here in the eukaryotic cells, the DNA will be in the nucleus. The DNA or the genetic material will be in the nucleus, okay? So in the beginning, we need to know what we mean by prokaryotes. So prokaryote cells carry genetic information in packages of DNA called chromosomes. So the chromosomes is the DNA package. Tamam? Most prokaryotes have only one circular strand of DNA. So one strand of the DNA, one circular strand of the DNA in the prokaryotic cells. Most of the prokaryotes have one circular strand, which is like this chromosome that is found already in the bacterial cell. This is an example of bacterial cells, where the DNA here is circular DNA, one circular DNA inside the cytoplasm of the bacterial cell. While the eukaryotic cells, the DNA must be in the nucleus, as we mentioned before, and the DNA is wrapped by the proteins called histone, and the DNA will bond and coil around the histone protein. The histone protein is made up of 8 separate units, and these units will join and let the DNA coil around these proteins. Together, the DNA and the histone proteins form the nucleosome, and chromosomes make the precise separation of the DNA during the cell division. Without the forming of the chromosome, the separation of the chromosomes will not be precise or sharp, okay? If the DNA is not in the chromosome structure, then the DNA will be like spaghetti, okay? A dish of spaghetti, so it's really hard for the cell to separate the DNA into two different cells, okay? As you can see here, this is the DNA, double helix DNA, okay, and this is the protein, which is called histone protein, is made up of 8 separate units, and these units will join to form the bud-like when the DNA coil around the protein. And the DNA and the protein here, when they are forming together, the structure called nucleosome. Look at the picture here again. This is the DNA. This is the double helix here. And the DNA with the histone protein, 8 separate units will bind to form the nucleosomes. The nucleosomes together will coil and then supercoil to form a chromosome. Or as we can say, this is a chromatid and this is another chromatid. These two chromatids will bind together to form the chromosome. As we can say that its sister chromatids will bind together in the centromere point to form a duplicated chromosome. So a DNA is considered to form chromatin, and which condense even more to form chromatids, which make up chromosomes. In the prokaryotics, okay, let's study the prokaryotes. Cells go through a process of cell division known as a binary fission. So we have a binary fission process, which is the cell division in the prokaryotic cell. In the cell division or the binary fission, which is asexual reproduction, will produce identical offsprings. The eukaryotic cell cycle having many phases. So let's start with the eukaryotic cell cycle. We have many phases. If you can read here, this is interphase, and here we have cell division. In the interphase cycle or part, we will have G1, S, and G2. So we have 3 phases in the interphase, and we have 2 separate phases in the cell division, what we call it M phase, okay? So we have G1 phase, which is the cell growth phase, and we have here S phase, which is the DNA replication phase, and here G2 phase, which is a preparation for the mitosis. In the cell division, we have the mitosis and cytokines. In the mitosis process, we have 4 different stages in the mitosis. Inshallah, we will take it at this lecture. So we have interphase, which is the time between the cell division, and it's have G1, S, and G2 phases. As you can see here, G1, S, G2 phases, and we have the cell division, the M phase. Let's study the interphase starting from G1 phase. In the G1 phase, the cells get larger, so the cell will grow. It gets larger and make new materials. There's many materials needed by the cell, so it will grow in this phase. In the S, which is synthesis phase, a new DNA, so we need to synthesize new DNA components. So G2 phase, prepare for the cell division. So we have three phases here, G1, S, G2. In G1, the phase it will grow and duplicate the material that is found in the cells, proteins, enzymes, anything that is needed by the cell, it will be duplicated here. In the S, synthesis, the DNA will be synthesized, the DNA will be duplicated, while in G2 phase, the cell will prepare itself for the cell division. Let's check now the M phase, which is the cell division phase. In eukaryotes, cell division occurs in two stages, mitosis and cytokinesis. Okay, so mitosis and cytokinesis. And the mitosis, it's mean the division of the nucleus, while the cytokinesis mean the division of cytoplasm. This is the process that happens in the mitosis. As you can see, this is the mitosis process, and this is the cytokinesis process. In the mitosis, the division will be for the nucleus, and in the cytokinesis, the division will be in the cytoplasm. For this point, we need to make a small review for the points that we take already in this first part. The first question is, how many strands of DNA do prokaryotes have? How many strands? For the prokaryotes, we have one DNA, okay? While nucleosomes are made up of what? Two things. What's the two things that is made of, the nucleosome made of? As we mentioned before, do you know what's the answer? Yes, okay, the nucleosome is made up of DNA and protein histone. Organize chromatids, chromosomes, DNA, and chromatin in order to form from smallest to largest. So we need to organize these components, chromatid, chromosomes, DNA, and chromatin, to form from the smallest to the largest, which is, yes, DNA is the smallest, then we have the chromatin, chromatid, and chromosome. Number 4, prokaryotes go through a process of asexual reproduction called, what it's called? Binary fission, that's correct. Number 5, what are the 4 phases of the cell cycle, which is G1, S, G2, and M. G1, S, G2 phase are part of interphase, that's correct. And what two phases make up the M phase? The M phase is made up of mitosis and cytokinesis. And again, the mitosis is the division of the nucleus, while the cytokinesis is the division of cytoplasm. At this point, I will stop. Thank you for listening, and make sure that you will watch part 2 of this lesson. Bye-bye. Have a nice day.